So now we're going to continue with photosynthesis, and we're going to study the Calvin cycle. And here in the Calvin cycle, we're going to take carbon dioxide from the atmosphere and through reactions fix it to form organic molecules like simple sugars. So your learning outcomes here are to describe carbon fixation and then to demonstrate how 6 carbon dioxide molecules can be used to make 1 glucose molecule. Okay, so building carbohydrates, in order to build the carbohydrates, this cycle is going to require energy because this is an uphill process. This is an endergonic process where recall that the free energy would be positive here. So positive free energy requires energy input rather than releasing energy. And so that energy is going to come from ATP, and that ATP came from that photophosphorylation we saw in the light-dependent reactions that occur in the thylakoid membranes. And this happens with both cyclic and non-cyclic photophosphorylation. Remember, in the cyclic one here, the photosystem is going to be short-circuited, so electrons are brought back after being excited at photosystem 1, so that we can create an even bigger hydrogen ion gradient within the thylakoid, so we can generate more ATP. In fact, 1.5 times more ATP are needed than NADPH for this process. So if we didn't short circuit and we just had non-cyclic, we would be generating a 1-to-1 of ATP to NADPH, so it wouldn't be enough ATP to drive those endergonic reactions that are needed. We'll see this in the cycle in a bit, and I'll bring it up again. So we need 1.5 ATPs for every 1 NADPHs. And what are the NADPHs for? They bring reduction potential to this process. This hydrogen that got attached to NADP came with a high energy electron, and this was generated back during the light dependent reactions of photosystem 1. So this NADPH is going to be the source of those energetic electrons and protons that we're going to be attaching onto carbon dioxide to make our sugar. So these light independent reactions, they can happen with or without light, so long as we have the reactants necessary, which include carbon dioxide and ATP and NADPH for the process. These were named after this Calvin cycle. It's named after Melvin Calvin, and this Calvin cycle is also called the C3 because the first product made after carbon is fixed is a 3-carbon compound. So when carbon dioxide comes in and gets attached to intermediates of the Calvin cycle, it immediately produces a 3-carbon compound. The key step here is to fix or attach carbon dioxide to a compound called ribulose bisphosphate. So that's the abbreviation for it. And ultimately, this compound is going to be here when it attaches, is going to form phosphoglyceracetate, which is a 3-carbon compound. So this is made up of 3 carbons. So that PGA. And this enzyme that's going to be used to do this initial fixing has a really big name, so they abbreviate it as Rubisco, but that Rubisco stands for ribulose bisphosphate, which means 2 phosphates, carboxylase oxygenase. Now, what function it has, so this suggests 2 enzymatic functions. This one is favorable, this one is not. Okay. So if you have too much oxygen, the enzyme ends up oxidizing stuff, and what we want to do is we want to reduce stuff, and we can do that by carboxylating our molecules. So this is the one that we're going to be following, but overall the enzyme is called Rubisco, and it's probably the most abundant molecule on the planet, since all plants on Earth and photosynthetic algae use this enzyme and the molecule ribulose bisphosphate to fix carbon dioxide. So overall, if we look at the right, we have our reactions, and these are going to occur in the stroma. So these are not going to be in the thylakoids of the chloroplast. So this is going to be in the stroma, or the fluid-filled area spaces surrounding those thylakoids. And there's 3 phases. Phase 1 is carbon fixation, which in the cycle we see right here. And you can see the carbon dioxides coming in, and they're going to be attached to our ribulose bisphosphate, that RUBP. And it's a 5-carbon compound with a carbon dioxide, so that comes in and forms 6 carbons altogether. And you split it in half, and that gives you a 3-carbon compound. Okay? And then from there, the next set of reactions is going to include a reduction. So here's where we're going to reduce the carbon dioxides coming in, or the molecule itself overall, by attaching hydrogens and electrons. So over here we have the reduction part of the phase, and there this involves the use of ATPs to help drive the reaction, and there's your NADPHs, which have that reducing power, and from there you're going to add the hydrogens onto the molecule, giving us a simple 3-carbon sugar called glyceraldehyde 3-phosphate. So overall that has to occur. And then while one of the goals is to end up producing sugar like glucose, the other goal is to help us regenerate. So that makes this a cycle. So we have to regenerate the ribulose bisphosphate so that it can go and fix another carbon dioxide. So here is your regeneration phase. It's important to note here that, as stated right here on the slide, that we have to turn go 3 times through this cycle to get one simple 3-carbon sugar called glyceraldehyde 3-phosphate. So you got to go 3 times to get just one. And it takes 6 carbons to make a glucose, so we need to combine 2 of these 3-carbon molecules to make one glucose. Okay, so we're going to actually need to go 6 times around, or go through the cycle 6 times to get one glucose molecule. 3 turns gives us simple 3-carbon sugar, and then those 2 3-carbon sugars going twice that amount. 6 turns gives us 2 3-carbon simple sugars that combine to make a glucose. So overall, basically we need 6 turns. So when you come back and you study this diagram more closely, and I'm actually going to cover it from another diagram here, but when we come back and study this diagram, you see it says 6 molecules. Okay? When I started talking about it, I only mentioned 1. Okay, so 6 molecules, 6 separate molecules of carbon dioxide are going to combine with 6 separate molecules of ribulose 1-bisphosphate. Each one of those is going to require the help of Rubisco, the enzyme. Okay, so let's take a look at this from a better diagram here. But keep in mind that we have to do 3 turns. So we have to do basically 3 turns around to give us just one glyceraldehyde 3-phosphate. Okay, and if we go 6 turns, then we're going to get 2 molecules of these, and those 2 molecules can be combined to form glucose. Okay, so coming to here's the same diagram, except we're looking at or the same process, but we're looking at it from a different diagram that shows the structural formulas. So starting right up here, this is the structural formula right here for ribulose 1,5-bisphosphate. And this diagram is going to show 3 turns, or 3 molecules of carbon dioxide being fixed at one time. The prior one was showing 6. Okay, so we're just kind of dividing everything by 2 here in the process. So 3 separate molecules come in, which would be similar to 3 turns of this cycle. So 3 separate carbon dioxide molecules are going to combine to 1 each on 3 separate ribulose 1,5-bisphosphates here. Okay, so 1 carbon dioxide and one of these, another one, and another one, another one, another one. So overall, when we do that, we're going to get 6 larger molecules, which are immediately, each one of those is going to be split in half. So if we look here, we count the number of carbons on ribulose bisphosphate. That's 1, 2, 3, 4, 5, and then you add a carbon dioxide, that makes 6. Okay, so right when each carbon dioxide combines to each of the carbon dioxides binds to one of those ribulose bisphosphates, you get a 6-carbon molecule that immediately gets split in half, right? So think about this. We're going to have When we combine the two, we're going to have 3 larger 6-carbon compounds, and each one of those gets split in half. What does that give us? A total of 6 molecules. And these 6 molecules are now that PGA, that phosphoglyceracetate. So this 3-phosphoglyceracetate. Why 3-phospho? Because if we call this carbon number 1, number 2, and number 3, there's a phosphate on the number 3 carbon there. Now what are we going to do here? We're going to energize this molecule by adding phosphates. So remember, we have 6 of these. Each one of them has 1 phosphate, and we're going to get now 6 ATP molecules and take one of the phosphates off of there and phosphorylate the molecule. So that's 6 of them for the 6 molecules here, giving us now 6 glyceracetate 1,3-bisphosphate. The 1 and the 3 because we have now a phosphate on the number 1 and a phosphate on the number 2. Now comes the reduction here. We're going to get 6 NADPHs. Each of them has electrons and a hydrogen to provide. And again, there's another enzyme there that, so all of these are enzymes back over here. So there's an enzyme to assist with this process here. And here we're going to attach our hydrogen with our electrons on there. So this is now our reduction because we're adding electrons to the molecule, right? And so what we end up getting here is a molecule that has an extra hydrogen on there. If we count the hydrogens earlier, we only have 1, 2, 3, 4. When we do the reduction for each of the 6 molecules, we're going to have 1, 2, 3, 4, and now 5 altogether. So our molecule is reduced. We have that extra hydrogen with those extra electrons. This is going to give us 6 glyceraldehyde 3-phosphates, and only 1 is going to exit. The other 5 are going to come through here to go through regeneration to get us back our ribulose 1,5-bisphosphate. Now notice that I said that the reason we have that cyclical photophosphorylation during the light reactions is to actually give us extra ATPs, because it's not a 1-to-1 for ATP to NADPH. If we just looked right here on this diagram, this is a 1-to-1. 6 ATPs and 6 NADPHs, but when we get over here, we're going to need 3 more ATPs to regenerate our ribulose 1,5-bisphosphate. So if we do some a little quick calculation here, 6 ATPs plus the 3 ATPs, that gives us 9 ATPs to 6 NADPHs for this process. And if I go and I reduce this ratio, then that's going to give us this 6/6 gives us a 1, and 9 divided by 6 is 1.5, which matches the numbers given earlier. So it's a 1.5 to 1 ratio of required ATP to NADPH to run this cycle. So what is overall the output of this Calvin cycle and carbon fixation? Glucose is not the direct product. Instead, a simple 3-carbon sugar called glyceraldehyde 3-phosphate is produced. And if we get 2 of those molecules, then they can combine to make glucose. Now, once the glucose is made, if it's going to be transported away from the leaves, it's going to be transported in the form of the disaccharide sucrose. So all we need to do is create the glucose and then use an isomerase to transform structural transformation, one of the glucose to a 6-carbon fructose instead, and then use that to make our sucrose. Recall from chapter 3, our biochemistry chapter, that sucrose is a disaccharide made of the 6-carbon fructose and glucose, which are isomers of each other. Also, when that sucrose gets transported to the right tissue, it may be stored in the form of starch, which is just a polymer of glucose. It can be stored for later use. Now, there is a connection between both photosynthesis and cell respiration. We've seen that if we think about the overall reaction. If I ignore the waters produced, right? So if I ignore the waters produced by the reaction of photosynthesis, we know that photosynthesis is 6 carbon dioxides with 6 waters. Usually it's 12 waters actually if we don't ignore the waters that are produced during photosynthesis. But this overall will simplify the reaction This is going to give us one molecule of 6-carbon sugar glucose and 6 oxygen molecules, right? So this can be oversimplified photosynthesis. We get our sugars, we get our oxygen produced. If we think back to our cell respiration chapter, what goes on in the mitochondria, that food molecule, that glucose, is going to go through aerobic respiration, which requires oxygen, and that produces our carbon dioxide and our metabolic water. So the two are basically opposites of each other. The waste product of one process ends up being the reactants for the other process, right? So over here we can put on our diagram at the top, this is photosynthesis. And this is cell respiration, going the other way, right? So that's what this is about. This last slide, a couple of slides here, that photosynthesis is going to use the products of respiration as starting substance. What are the products of respiration? Carbon dioxide and water, H2O. And respiration, which happens in the mitochondria, is going to use the products of photosynthesis, which are glucose and oxygen. Products of photosynthesis are these. Those are used by cell respiration. Looking at these last 2 points here, the production of glucose from glyceraldehyde 3-phosphate, which is the simple 3-carbon sugar, actually uses an ancient part of the glycolytic pathway. If we went back and we studied glycolysis, you would see this compound mentioned in there as one of those intermediates for glycolysis. So imagine we're kind of running in reverse from glycolysis to go and make our glucose molecule. And if we also look at how the electrons are handled within the membranes, there is an evolutionary relationship for those electron transport proteins that help us produce ATP. So they're evolutionary related, those electron transport proteins that we see in chloroplasts and in the mitochondria, which is pretty interesting and cool. So if we look at this diagram, it overall does show the relationship here. If we see arrows leaving, that's a product of the process. For example, here we see the light reactions when we split water produces oxygen. That's used as the final acceptor in electron transport over here in the mitochondria. If we look at the products of the Calvin cycle, which are the dark reactions or the light independent reactions, is glucose, and then glucose in the cytoplasm goes through glycolysis to produce pyruvate, which enters the Krebs cycle over here. Now, what are products of the Krebs cycle? Carbon dioxide. Well, carbon dioxide is used by the Calvin cycle to make glucose. And what's a product of electron transport in oxidative oxidative respiration? We have one of our products is that metabolic water, which is used by the light reactions. That's where we split the water to make oxygen. So you see the products of one are used as reactants in the other process. So there's a connection between the two.